Välkommen till podcasten Psykedelisk vetenskap. I det här avsnittet pratar jag och Gustav med Attila Szabo, som är ledande forskare inom kopplingen mellan psykedelika, biologisk psykiatri och immunologi.
All right. Hi. Welcome.
Hi. Thank you very much for having me.
And welcome, Anja.
Thank you. Hi.
Yeah. Great to have you here. So, if you can begin about talking about your background a little bit.
Of course.
Yeah.
So, I'm basically my formal background is in biochemistry. So I did my first master's degree in biochemistry. I graduated as a biologist back in Hungary. Around that time, this program was basically 5 years program, so we didn't have bachelor's and master's. I did it in biology/biochemistry, and then I did my PhD in immunology, and this is where when I was actually exposed to serotonin research, so to speak. So I kind of started reading about serotonin. My original background is in inflammation research, so I was mostly interested in inflammation and the regulation of innate immune responses, which are connected to inflammation mostly in humans. And after doing a couple of years of PhD research on this topic, around 2010, I started to read about psychedelics, mostly these early papers that were published in the 50s, 60s, before the CSA, the Controlled Substance Act. So before, in the US, it was basically shut down.
Yeah.
And I found those papers pretty fascinating. And with the background in inflammation and serotonin, signaling and all of this stuff. And, these psychedelics, most of the psychedelics that we are talking about as psychedelics
they are
serotonergic.
Yeah.
Exactly. So it got me really interested, and I wanted to do something at least at the level of scientific research. But obviously beforehand, I was also interested in psychedelics in general. And in the late 90s, early 2000s, I was also reading books by Hoffman, Terence McKenna, all these big from Ott, and many books on ethnopharmacology, ethnobotany. Some of them more scientific, some of them less scientific. I was interested also in this kind of counterculture or revival of a counterculture, if you like.
Right. The rave scene of the late 90s.
Yes, absolutely. So it was kind of a good constellation of interests in both in the the cultural aspects and also the scientific aspects. During my PhD years, actually in the last year of my PhD and before I started working on my first postdoc project, I met with Professor Defrechka, who was at the time a chair at the Department of Psychiatry at the University of Debrecen, which is my alma mater in Hungary, in East Hungary. And at the time he just came back from New York. He spent 17 years in the US. He was a resident psychiatrist at Mount Sinai in New York. And he was involved in lots of research, so he had a strong research background, mostly with regards to antipsychotics and schizophrenia. But also he was interested in psychedelics, specifically dimethyltryptamine and hallucinogenic tryptamines. And he offered a collaboration. So I would say it's out of mere serendipity, I ended up attending one of his lectures on ayahuasca and DMT. And then we had a talk. And the chemistry was really good. So we almost immediately started to, figuring out ways of future collaboration.
So this is when you started, getting professionally into psychedelics?
Exactly, yeah. Exactly. So that was basically the foundation stone, I would say. It was around 2010, beginning of 2010. And then ever since, my work has been focusing on the immune effects, mostly the immune effects and the cellular molecular effects of the psychedelics on inflammatory pathways, stress pathways, mostly on preclinical models, in vitro studies and animal in vivo studies. But recently I've also started to collaborate with other nowadays well-known groups who are pretty big now in the field, including Jan Ramaker's group in Maastricht University, and also multiple groups at Imperial College London. For instance, one of is that is headed by David Eritzoa.
Yes.
And those studies are mostly focusing on identifying potential biomarkers of psychedelic effects, inflammatory and immune biomarkers, psychedelic effects in humans.
Using different methods then, so not in vitro studies or?
Correct, yeah.
Okay.
So we are talking about, in this case, we are talking about mostly collection of blood samples and saliva samples. Looking into specific things or biological indicators that can indicate or predict therapeutic effects in different disorders, from OCD to depression. And these are many. So my focus has been kind of shifted from DMT and 5-methoxy-DMT to psilocybin, and most recently ketamine. So now I'm also interested in non-serotonergic, non-classic psychedelics as well, which are very promising as well, by the way.
So you're looking at biomarkers of different mental disorders related to inflammation. Can you give us some introduction to the field of psychoimmunology and what is the relationship with, between inflammation and psychiatric disease?
Absolutely. It's a pleasure. This is my other specialty. So my main specialty at the University of Oslo is psychoneuroimmunology. And where I actually my projects are focusing on psychotic and mood disorders, schizophrenia spectrum and bipolar 1 specifically, and more recently autism spectrum disorders. Here we're trying to find connections between brain behavior and the immune system, or dysregulation of the immune system. Many of these disorders, especially in case of major depressive disorder or schizophrenia spectrum disorders, it's been more or less well known. It's been actually an established paradigm in psychiatry that there is an inflammatory signature, at least in a subgroup of patients. This means that in case of patients with MDD, about 52% of the patients have elevated levels of some of the inflammatory cytokines when it comes to baseline levels.
And what are cytokines?
Cytokines are basically the language of the immune system. So these molecules are used by the immune system to communicate with each other. So cells of the immune system use it as kind of a language. And also they communicate with other tissue cell types, such as endothelial cells, like cells of the blood vessels, or brain cells even. And then we see these receptors which can detect these cytokines or understand the language, speak the language of cytokines. We see these receptors also expressed on brain cells, like neurons in the prefrontal cortex, here in the anterior part of the brain, which is responsible for higher order cognition and social cognition. And it's basically, working. It's a bidirectional communication. So brain cells, glial cells in the brain can also produce cytokines, and that signal can be picked up by the peripheral immune system. So psychoneuroimmunology is basically a field within psychiatry and behavioral sciences and immunology, which is trying to understand the nature of this communication and its role in health and disease.
It's very interesting, this relationship between the immune system and psychiatric disorders that you mentioned. Do we know, or do we have any theories about the direction of that relationship?
So if you're asking about the error of causality, that's hard to tell. So there are some instances where we can say that there are some causality involved. But this is like one of the aspects of psychoneuroimmune research, which is under development and research. But there are definitely some well-documented phenomena that we know of. For instance, let's say someone is subjected to severe psychosocial stress. Stress in general, in the psychosocial way, can for instance induce an inflammatory response in the body in healthy individuals, otherwise completely healthy individuals. And by that I mean someone who doesn't have any diagnosis, right? Any clinical diagnosis, neither immunological nor psychiatric or psychological diagnosis. So, and on the other hand, ongoing infections or inflammation can lead to changes in behavior. Best example is, let's say, someone is having a viral infection, COVID or flu, common flu or just a common cold. And activation of the immune system and response of the immune system to this infectious agent is also associated with the production of these cytokines, or a class of cytokines. This is obviously a very complicated story, so I'm using cytokines as an umbrella term. Of course, certain disorders or certain infectious agents are provoking different types of cytokine production. But in general, say someone is having an infection, elevated levels of these cytokines can act on the brain, certain parts of the brain, which will basically change the behavior accordingly. So the reason why you feel weak and shitty, so to speak, during an ongoing flu infection is not because of the virus. It's actually because of your immune system. Right. Cytokines are making you sick, quote unquote. This sickness even has a term in psychiatry. It's a textbook term. It's called sickness behavior. And we've known about this for decades. So it's a long-known phenomenon.
So are there like signature types of inflammation that correlate to distinct groups of mental disorders?
Yeah, so this is also a very good question. There are at least a few cytokines that have been associated with certain types of disorders. For instance, in case of specifically therapy-resistant major depressive disorder, interleukin-6, which is one of these cytokines. Elevated levels of interleukin-6 seems to be very important. IL-6 is both a state and a trait marker in MDD, which means that you can see slightly elevated, so-called chronic low-grade elevated levels of interleukin-6 in a large population of patients. It's about half of the patients, or 40% of the patients. And also you can detect elevations of IL-6 during depressive episodes. In case of effective treatment by an SSRI or some kind of antidepressive treatment, MAO inhibitors, mostly SSRIs, as the symptoms start to improve, the level of IL-6 starts declining. So there's this super exciting association between these things.
But there again, I suppose there could still be a problem of causality.
Of course,
yeah.
So the thing is that we will not understand causality perfectly until these controlled trials are done on large populations with different disease cohorts, preferably using some kind of intervention, such as a pristine, pure, and anti-inflammatory drug / an antidepressant, or the combination of the two. There have been many smaller studies using this type of protocol, and I've seen a couple of interesting papers published on it already. But so far, at least, it would be hard to say anything about the actual causality.
Right.
Nonetheless, very interesting.
Very, very interesting.
Yeah, definitely.
Maybe this is a good time to bring up psychedelics again, and the effects of psychedelics on the immune system, and how that might affect, or how that might help us understand, the effects of psychedelics as an antidepressant, or.
Yeah, absolutely. So the thing is that we've already talked about serotonergic psychedelics, or the so-called classic psychedelics, which exerts their effect on the body, on the brain and the body via serotonin receptors. It's not entirely true, because the term serotonin receptor is also an umbrella term. There are many, many subtypes here. And we know that only certain types of serotonin receptors are involved in the actual psychedelic action. On human physiology, for instance, when it comes to the mind-altering effects, we know that at least one of the receptors, serotonin 2A, is heavily involved. And we also know that there are some other types of receptors, like serotonin 2B and serotonin 1A, which are involved in at least some effects on mood, personality, and cognition.
Right.
So when it comes to immune effects, one important thing to mention is that immune cells also express serotonin receptors. So they have these sensory organs, so to speak, so they can see and react to serotonin, like blood circulating serotonin. Now, another thing here, which I should mention before I actually delve deeper into the topic, is that to talk really, speak really very briefly about serotonin. So serotonin, from an evolutionary perspective, is a very ancient molecule. Serotonin does have lots of multiple roles besides cognition. and mood regulation. It has a role in regulating appetite, regulating the body temperature, core temperature, blood pressure, and many other things. And one of these many other things is the regulation of immune responses and inflammation. It's also kind of a well-known, well-documented phenomenon in the literature. There have been many papers published on it in the 80s, 90s on the immune role or immunoregulatory role of serotonin, right? But when we are talking about serotonin, we are talking about this ancient molecule which acts on all the receptors. Right? When we talk about serotonergic psychedelics, I just mentioned that based on the given psychedelic, only a couple of these receptors are kind of being engaged or ligated by the psychedelic. Yes. So initial researches on the potential anti-inflammatory and immune modulatory role of psychedelics were directed towards or focused on these serotonin receptors, 5-HT2A included. For instance, Chuck Nichols' lab done landmark studies on it back in early mid-2000s. For instance, they also used a mouse model of asthma where they tested the anti-inflammatory role of DOI, which is a phenethylamine, is an amphetamine derivative. Those were the first kind of steps, so to speak, in the understanding of psychedelic effects on the immune response. After this, I got really intrigued by this, and I really wanted to kind of follow up on these researches, but do something with human immune cells. Thanks to my access to the lab infrastructure at the Department of Immunology at the University of Debrecen, where I was working at the time, we had a quite stable, robust pipeline where I had the ability to test tryptamine effects on multiple cell types, human immune cell types. And thanks to my background in immunology and in inflammation biology, I knew how to provoke inflammation, how to frame or design an experiment in a proper way, and how to test this psychedelic, this tryptamine psychedelic. And then what we found was that DMT was also very just like the phenethylamine DOI, the classic tryptamine serotonergic hallucinogen also possessed the significant immunomodulatory potential. It appeared to be a very potent anti-inflammatory agent. And we managed to identify another receptor which was basically involved in this action, in this anti-inflammatory action. It's called the sigma-1 receptor. So we published it for the first time back in 2014, like about 10 years ago, the paper on the anti-inflammatory role of DMT and 5-methoxy-DMT through sigma-1 receptor. And also partly through 5-HT2A, but that had been known already, right? It was part of the paradigm. So when we are talking about the anti-inflammatory role and immunomodulatory action of psychedelics, most probably the two most important and interesting receptor types are 5-HT2A and sigma-1. But obviously it's still an unexplored field, so obviously there are like large uncharted territories here. And when we are talking about the immunomodulatory role, most of the available literature is preclinical. This is mostly in vitro and animal in vivo studies. And there's only like small number of small cohort studies which screened for maybe one or two or like a low number of inflammatory markers. And also observing similar patterns, confirming these initial or these first papers, the in vitro findings.
So how would you like to design a study where you could actually see this in live humans, say?
So the first thing is that what you can do is, and probably the easiest and ethically least problematic approach is to recruit some healthy volunteers who have preferably already had some experience with the given psychedelic, right? There are protocols for this that I'm not entirely familiar with at the moment, since I'm kind of stronger in the preclinical field. So I've never been, I'm not a clinician, so I have not been involved in recruitment procedures, or. But I do know that there are obviously parts of protocols or actual protocols on it, how to recruit properly. And I also do know that initial or previous experience with the given psychedelic is preferable. So healthy volunteers, and then obviously based on the PK, pharmacokinetics and pharmacodynamics properties of these drugs. So based on the knowledge on through which receptors it's acting on, or which receptors it has any agonistic effect on or at, and also based on the timeline of the drug, so to speak. So the pharmacokinetics properties. You design the study, so you design the treatment period, and then the easiest one is basically just to take samples, let's say blood or saliva samples at baseline, so before the actual treatment or actual administration of the drug or the substance, and after the substance.
And you would look at cytokines of certain kinds that are associated with mental disorders?
Sure.
Yeah.
So in this case, like the time window is designed in a way that, you need to know which cytokines are looking into. Some of the cytokines are upregulated and produced maybe 3-4 hours after the administration, after any kind of let's say exposure to some infectious agents. You can also provoke inflammatory response with LPS injections. It's basically isolated from bacteria. You can administer it intravenously and cause a systemic inflammation, like a mild inflammation. So after deciding on which cytokines are you looking into, you basically design the timeline and you collect, let's say, blood and saliva like 3 hours after administration, 6 hours, and 1 day after administration. And then you can see, based on the kinetics, you can see also the kinetics of the cytokine response. And then you can shortlist those cytokines that are involved, as you said, the pathophysiology of depression or schizophrenia or whatever is your main focus or foci are. So basically, that's the easiest way. And then you can also include an interventional aspect, where you can use a placebo-controlled framework and let's say use some NSAID, some anti-inflammatory drug, which is non-invasive, NSAIDs, non-steroidal anti-inflammatory drugs such as aspirin or paracetamol are preferred in this case rather than, let's say, steroids, which have a massive effect on the immune system, can cause immune suppression. And then you can compare the effects of these already existing anti-inflammatories with different doses and different treatment regimens with the given psychedelic or psychedelics you are testing.
So are there any maybe more physiological immunological disorders that are associated with serotonergic or the cytokines that might be affected by serotonin activation? Yeah,
many. So this is actually one of the probably the hottest part of this type of research. So autoimmune disorders is a large family,
right?
Consisting of many disorders. It's in the hundreds, probably, including all the subtypes and all the different forms, which include also polymorphisms, like genetic predispositions. So there are many of these, and there are also some very promising candidates when it comes to psychedelic-based treatments, psychedelic-based therapies.
Like what?
Yeah, for instance, I would say that knowing the background, the cytokine signature, and the type of immune mechanisms involved, without going into the details, there are some very promising ones. For instance, rheumatoid arthritis, I would put that in the top 5, and multiple sclerosis. And probably also SLE, however, systemic lupus. However, that's a bit complicated because SLE is typically a systemic autoimmune disorder which affects multiple systems. Also, the so-called adaptive immunity, part of the adaptive immunity, which is probably just partly affected by psychedelics. But for instance, designing a study protocol on SLE, at least with a small cohort of patients with SLE, would also help to clarify, the effects of psychedelics on the adaptive immune response, not only the innate immune response.
And it might be a little bit easier methodologically because, just considering ethical approval and stuff, it's typically easier to do studies on healthy participants than healthy participants.
Absolutely, absolutely.
So have there been any studies like directly showing effects on inflammatory autoimmune disorders in humans with psychedelics?
Yeah, not that I know of.
Yeah,
it's not in humans, at least. So I know that currently there is a recruitment for a small, for small RA rheumatoid arthritis study in psilocybin in London. I can't unfortunately talk about the details because I signed an NDA. But it's ongoing. It's in the recruitment phase, and birds are chirping that there's another one which is will be in the recruitment phase later this year. This is 2024 in the US. Also, I can't disclose any more details about that.
Very interesting. We will have to look out for those papers in the future.
Yeah, hopefully soon enough. Yeah.
So we were speaking about also like psychiatric disorders and inflammation. So I'm a psychologist, so of course I'm biased towards the like psychological and behavioral mechanisms that perpetuate mental distress. Could you speak a bit about that, like the relationship between the biological inflammatory causes of mental illness and also the psychological and behavioral ones?
Of course. So probably a good example here would be schizophrenia. For instance, like in case of schizophrenia, we have in psychiatry we have this, in clinical psychology we have this so-called two-hit model, which means that in order to someone to develop schizophrenia, the person needs to have genetic predisposition. That's one hit. So if the genes are there, so-called genetic loading is high enough, then it's not enough by itself. So it's not enough to develop the disorder. There needs to be a second hit, which is usually an environmental effect. And this environmental effect can be anything. From intrauterine infections like maternal infections, also like smoking, being grown up in a family where you are surrounded by people who are smoking, like exposure to toxic material, also combusting fumes, chemicals, also like severe infections in childhood. So this is most probably true in case of many psychiatric disorders with neurodevelopmental aspects, such as autism and schizophrenia spectrum. In other cases, probably, I'm not an expert in psychology, but I do believe that we obviously can't blame everything on the organic causes. So obviously, psychosocial factors are really important. This we know from twin studies of at least schizophrenia and mood disorders, where the same type of genetic loading and also more or less same exposure to similar conditions does not necessarily lead to the development of symptoms in both twin pairs, both twins.
Right.
So probably both are important. If you ask me, what I'm coming from a biological psychiatric paradigm, like I'm approaching the topic from this perspective. And it seems, based on what I've read and what I've heard in conferences, this whole inflammation thing and whole infection and immune modulation thing definitely shows the importance of environmental effects, but also genetic predisposition.
Right.
So
one thing that's quite hot in my field, in like clinical psychology, is like talking about like psychological trauma, psychosocial distress, and that effect on inflammation. And so
it is also known. Yeah, I've seen a couple of papers within that field as well, and I have actually a couple of colleagues who are working specifically on this topic. So early adverse events and childhood trauma and their effect on circulating cortisol and circulating inflammatory markers in different disease populations. Mostly, I think those are bipolar 1 specifically and schizophrenia, so psychotic disorders in general, There's definitely, so it means it seems that early childhood adverse events do affect immune regulation or immune dysregulation later in life, definitely contributing to it.
So that can be an additional hit on the pathway to developing, say, schizophrenia.
Absolutely, absolutely. But it's probably this is what we call the second hit. So this is the environmental effect, right? Or part of the environmental effect. And again, it is most likely that it's not necessarily happening in anyone due to, again, like genetic load, right? So someone who is like kind of genetically more resistant to the given disorder because of the lack of predisposing factors, or because of like protective gene variations or variants, will unlikely to develop the disorder. But definitely it's important. It's a very important mechanism.
And what about depressive and anxious disorders and the pathway of inflammation to those like presentations? Yeah,
so anxiety, unfortunately, I'm not really familiar with the anxiety field, so I can't really comment on it. Depression slightly more. So as I said, in slightly less than half of the patients patients with MDD, major depressive disorder, display some kind of immune abnormality. And this immune abnormality is most typically elevated levels of the so-called interleukin-6 that I mentioned, and some other markers such as CRP. But CRP, C-reactive protein, that's a typical inflammatory marker which is routinely used in clinical studies. But nowadays people are getting a little bit like less enthusiastic about it because there's like huge heterogeneity of CRP levels. So IL-6 seems to be the best candidate in that regard. And again, as I said, IL-6 seems to be a good state and trait marker, because again, it seems to be like dysregulated both at baseline and also during episodes of depression. And
So back to psychedelics, what are some other things you've been working on recently? You talked about some of your earlier research and some of things that are going on, but what about the things you've been publishing in the last years?
Yeah, so most of the things that I've been publishing about were small cohort human studies in various settings. So some of these studies were focusing on the effect of smoked or inhaled 5-methoxydimethyltryptamine, the bufo, this toad secretion, and also synthetic 5-methoxy, and comparing the two.
Interesting.
Yeah, on immune functions and on stress response in humans and other stuff, like similar stuff. Behavioral.
So that's very very rare, like directly comparing like a naturally occurring compound with a chemically produced one, if I understand it correctly.
Yeah, absolutely. And
what did you find?
So these studies were spearheaded by Malin Uthaug.
Yes.
She's a well-known specialist in the 5-methoxy DMT field, and I think most of her projects, at least those that I've been participating in, were anchored to Maastricht University. But I know that she's associated also with Imperial College and also the university in Australia. I don't remember which one. I'm sorry. But anyway, it doesn't really matter. So in the Maastricht, Jan Remaker's group-based studies, so we looked into immunomodulatory potential, but also the behavioral modulatory effect of 5-MeO-DMT, and then there are actually many different administration kind of routes. It can be first and foremost, obviously, smoked 5-MeO-DMT, but can be also intramuscular injection. And also we can also use, instead of using the secretion, bufo secretion, we can use purely synthetic 5-MeO-DMT. And it seems that the purely synthetic one seems to be as effective as the natural one.
So there is no difference really?
I don't know any important differences when it comes to behavioral outcomes, which is very important from a psychotherapeutic perspective. And I haven't performed any actual biomarker analyses on this type of comparison. But what they've done, and I think already published at least one paper on it, maybe two. There are definitely huge overlaps in the behavioral outcomes, which is very important because from an ecological and conservation biology perspective, this is like might have a major impact.
Yes,
because obviously, like
they are kind of threatened, right? The buffalo.
Yeah, absolutely.
Yeah.
It's a protected species, and the populations in Mesoamerica is like definitely threatened now.
Yeah,
and obviously, coming up with first and foremost synthesizing good quality standardized batches of 5-methoxy-DMT is not not complicated and not expensive. And let's say if we can achieve the same effects and same psychotherapeutic effects, it would be way better from a conservation biology perspective and ecological perspective. So
it's great to hear that you're doing that direct comparative research. So that's a good argument for not harming these toads.
Oh yeah, absolutely, absolutely.
As if we needed even more arguments.
How far along are you in the study? Have you collected all the data, or is it ongoing?
So this comparison study on the biomarkers, it's planned. So I haven't started analyzing because we have just published another one that I haven't mentioned yet, and then we are collaborating on yet another study right now. Malin just collected all the samples in Canada. And when I'm talking about these collaborations with Maastricht University, it's always a bit complicated because I work with both blood samples. For instance, in our latest project with Natasha Mason, we used blood samples. So we looked into blood circulating biomarkers. While most of the studies that I've collaborated with Jan and Malin, those were performed using salivary assays and saliva samples, which is kind of nice. It's good, and there are actually advantages and disadvantages in this case. A huge advantage of salivary sample collection is it's non-invasive. It's easy, right? And you can collect it before. It's just a spit, right? A sputum, and you can even collect it during or right after the ceremony.
Right.
So while using a needle to collect blood, it's obviously impossible. During an ayahuasca or an 5, especially during a 5-MeO, ceremony. So, but on the other hand, when it comes to advantages of for instance, using blood samples, is that you can use much finer and like smaller differences in some markers. You can look into way more markers at the same time, and you can run the sample on a way more intricate analysis pipeline. So again, what you can get out of the same amount of sample is way more data and more reliable data when it comes to human physiology. So actually what's going on in the body when someone is exposed to the given psychedelic.
Very interesting. So in addition to all of this research you've done, you also have a master's in philosophy. I was wondering if you could talk a little bit about that. What are your philosophical interests?
Yeah. So it's a very old story and long story. I had always been interested in philosophy since I would say high school. It's just first, especially due to the pressure of my parents, I had to choose something that I can live off of. Some like more useful professions. Yeah. Exactly, STEM, one of the STEM subjects. So I went for biology because I was fascinated by biology. Actually, equally fascinated by psychology and biology. But then I ended up doing biology because it was something that I was really interested in. Nonetheless, I loved courses in evolutionary psychology and animal behavior. So that was really good. And I think it's incredibly helpful when it comes to psychedelic research. So I highly recommend anyone, who's planning to embark on this journey of psychedelic research to do something in either psychology or biology. Or both. If someone is, let's say, a psychology student, it doesn't hurt to just pick up a textbook in biology and then, read something about animal behavior. It's super fascinating and can help a lot to understand the bigger picture. And anyway, I ended up studying biology and then specializing in biochemistry. But in the meantime, I felt that it wasn't enough. Like it was not I didn't feel like my understanding was complete of the universe. I know it sounds stupid and a little bit of cliche. You can never have a complete understanding of the universe, right, or reality. But I felt that philosophy is something that could help me. I've been always fascinated. I had been fascinated by all these different philosophy books that I was reading in high school and in the early years during my undergrad and grad studies in biology.
It's very useful to be a scientist, right? If you have some sort of knowledge in philosophy.
Absolutely. Like a formal training, at least in logic and in philosophy and history of science, is absolutely instrumental. Yeah, it can be super useful.
And was this some of your interest when you were studying philosophy?
Initially, yes. So first, actually, I also did, by the way, a 5 years training in philosophy. So not bachelor's, master's, but a 5 years complete program in philosophy. And so initially I was focusing on actually logic and philosophy of science, specifically Kuhn and the structure of scientific paradigms, right. And Feyerabend and what is science, what is not science, what is epistemology, how can we what I was particularly interested at the clashing point of paradigms and how we formulate hypotheses and how can we actually test them without making like fundamental mistakes in data interpretation. So it was super cool and I really loved it. However, after 2-3 years during my philosophy studies, I fell in love with the phenomenology. I think I can thank Kant. So I became a Kantian for about a year or two, and then also I was partly kind of running away from Hegel. I didn't particularly love Hegel courses, so I tried to get as many credits from Kant instead. Yeah, exactly. So and then I started reading hardcore phenomenology,
like Husserl,
Heidegger, mostly Husserl first, like classical phenomenology. And Heidegger, but then later on, like a bit more modern approaches in phenomenology, especially this, the paradigm of embodied cognition,
right?
And also like Dan Zahavi's books, I truly enjoyed. So definitely was something that I was fascinated by, and at the same time I also did some courses on Jung, Jungian psychology. So all of these had a major impact on me. I was interested in the philosophical aspects and the mind, the consciousness, philosophical aspects of psychedelics.
Yes.
So
I'm wondering if your interest in psychology and philosophy and phenomenology, has that influenced your professional work in psychedelics in any
Definitely.
way? Yeah,
definitely.
So could you give some examples of that?
Yeah, I would say definitely was steering me towards psychedelic studies and research all the way. I mean, I can't really tell you any like actual examples when I realized that this is what I want to do. It's just happening. It was a process. It's a long process, but it started in my late teen years and got culminated my studies in philosophy, and then also like doing research, hardcore research on psychedelics. So actually choosing this as one of the main tracks of my scientific career, which is, by the way, back then it was not cool yet, right? And we were talking about 2010, 11.
Yeah,
so it was heavily stigmatized, right?
Yes.
So it was
happening a little bit in some places, but it wasn't acknowledged or. Yeah.
Correct. And it was not. I mean, that's kind of a euphemism to put it that way, because it was like so heavily stigmatized that, for instance, for my first study with DMT and 5-methoxy-DMT, I had to wait. I had to constantly struggle for about 4 years to get all the paperwork done and to get all the permissions from the police department and the state of Hungary and the EU approvals for all the work, even for just small in vitro studies. So not human use, not animal use, just in vitro.
So was it difficult to get a hold of the substance, or researching it, or even in like cell models?
Yeah, both. So again, like cellular models, in vitro models, small amounts. Just so you know, so when you want to work with substances like this, or back then at least, it was first you have to apply for a so-called purchase permit. So after getting purchase permit, basically allows you to buy a certain amount of stuff and use it for a well-defined type or experiments. So one or several studies with a well-defined focus. And you have to define how much you will use and what substance we'll use as part of the purchase permit. And then this allows you to buy it from a provider, from a supplier. But you also need an ethical approval, which is a bit less complicated when it comes to in vitro studies, cell studies, but nonetheless you need it. And then finally you also need one for the import. So you need an import permit, which is probably the toughest, because even though you have all the permits done, if you don't have the import permit, you can't import the stuff. So unless you have a supplier within the state or the country where you live, which is very unlikely, was unlikely back in
2010 in Hungary, there weren't many 5-MeO DMT chemists.
Like, we are talking about a chem grade 5-MeO DMT, right? So it has to be pure, it has to be sterile to be able to use in. So it was complicated, to say the least. But anyway, I was just too motivated. And fortunately, my colleague and mentor, Ed Frechka, who was also became a good friend of mine, he was pushing very hard, and he used all of his network, all of his capacities in the US and in Europe. So we finally managed to get our hands on a tiny amount of DMT and 5-MeO DMT.
Oh, wow.
And then that was enough for the first 2 studies. So I was very happy.
Yeah, that's great.
Yeah.
Yeah. So we're about to end, but I was wondering, we can end with a very big question. Like, since you have this interest both in like the biological, materialistic side of psychedelic research, and also a strong interest in phenomenology, what can psychedelics tell us about like the mind-body connection? And
wish I knew the answer.
Yeah. But maybe talk about some of the aspects you're interested in, or your thoughts about that. Yeah.
Basically, this is exactly, so as you formulated it, this is exactly what I've always been interested in. And my scientific career, psychoneuroimmunology, is also basically this, like trying to answer this question, right, from through the lens of science, data-driven science and empirical science. Definitely. As I say it, psychedelics offer us a super useful and unique tool, or tool set, if you like, to tap into realms, look into realms that are otherwise closed off of our cognition, and can definitely must be instrumental, in catalyzing understanding of this whole mind-body phenomenon and mind-body problem. And I wish if like David Chalmers was taking like larger doses of LSD and come up with a new revised version of his hard problem of consciousness. But definitely, the way I see it is definitely something that could be helpful in that regard. And I mean, probably it's impossible to tell when we will make any progress here. But getting actual results is actually not that important than being part of the movement and like making progress, and then doing our best to explore these uncharted territories. Yeah, I
think that's a perfect way to end, right?
Yeah.
Yeah. All right. Thank you so much.
Thank you very much. It was a pleasure.
A very good conversation. Yeah.
Thank you very much.
Tack för att du har lyssnat. Vi som har gjort podden heter Anja Winter och Gustav. Podden är producerad av Nätverket för psykedelisk vetenskap, men åsikterna som uttrycks är intervjupersonernas egna och representerar alltså inte föreningen i stort. Läs mer om vår verksamhet och bli medlem på mpv.se. Musiken är gjord av Manne Skavenstedt.